Bismillahirrahmanirrahim. Today we have to talk about the nutrient acquisition and transport, the long distance transport, which we are already continuing. So today is the second part of that, which we will talk about. In the first, uh, we already discussed about the xylem transport, where we discussed about the composition of xylem sap, then xylem loading and unloading, uh, then effect of transpiration rate on the uptake and the translocation, effect of transpiration rate on the distribution within the chute. So today we will talk about the phloem transport, and phloem transport is very important, like the xylem transport, because it helps the nutrients to move into the different parts of the plant body and different organs. So basically, uh, as we know that the xylem movement of sap in the xylem, or the movement of the ions in the xylem, that depends on the root pressure and the transpiration rate. Uh, In phloem transport, it depends on the pressure flow hypothesis. So that was actually presented in 1930. So what is this pressure flow hypothesis? It's very simple. It is based on the osmotic adjustments. We know that the where the osmotic potential is more negative, uh, due to the high concentration of salts, either these are inorganic salts or organic salts, then the fluid will move towards that part, because the water or the fluid move from higher potential toward the lower potential. And the concentration of these solutes that decrease the potential of the water, and so that's why that fluid move from higher potential towards a lower potential. So that all the phloem transport is based on this pressure flow theory. So this type of pressure-driven mass flow in the phloem differs from that in xylem in three important ways. Uh, in the xylem, we have observed that they are mostly dominant in the inorganic compounds, because the nutrients they take up ions are inorganic compounds from the soil, and they just transfer it into the or load it into the xylem. So that's why the xylem sap has mostly inorganic compounds, but the phloem they have dominant organic compounds or solutes in the phloem sap. Uh, and the phloem is not a dead sieve, it is a living cell, and the transport takes place in this like living cells. The unloading of solutes at the sink plays an important role. Unloading of solutes, because uh, the phloem is also, although the in xylem the unloading also occurs, but in the phloem, because it has many organic and inorganic solutes, and it is not only uh, transporting uh, the ions and compounds into the upper parts, but also towards the roots. So the unloading uh, is very important because it has many organic compounds and many inorganic ions as well. The unloading will determine or will define the composition of the phloem and the movement of these compounds into the phloem. Uh, what is the composition of phloem? If we look at the concentration All essential elements are present relatively higher concentration in the phloem. So in the previous slide, we have mentioned that the phloem sap is dominant in the organic compounds, but it doesn't mean that it uh, do not have or it doesn't have the inorganic ions. Phloem is mostly dominant in inorganic ions because we know that the most of the ions they are mobile and they are recirculated in the phloem. So once they are loaded into the sink, as most of cases is the leaves, so it is retranslocated or recirculated in the phloem because they are mobile. So that's why the concentration of ions in the phloem is more than the xylem. Although they are dominant by the organic compounds, but even the ion concentration is also more in the phloem. Sucrose, organic acid, hormones, amino acids, secondary metabolites, RNA. So they are also present in the phloem sap, and that makes more dominant organic compounds in the phloem. If we just analyze the composition of uh, xylem and phloem, or if we just compare the concentration, uh, the phloem, the pH of phloem that is 7.9 to 8, and pH of xylem is 5.6 to 5.9. So this is a major difference in the pH uh, and pH of phloem and the pH of xylem. Uh, if we look at the sucrose concentration, uh, the the concentration of sucrose in the phloem that is 145 to 168 milligram per liter, which is uh, not in the xylem. You may find some, but it's not the organic compounds. They are not present in much quantity in the xylem sap. Of course, overall, the dry matter of phloem is also more than uh, the xylem. Then the amino acid concentration you can see here, 10,808 microgram per liter, and which can be seen very low in the xylem sap. Then we have ammonia. Again, you can see there is a very, very much difference. And the highest difference, perhaps you can see in inorganic ions, so that is potassium. So basically, the concentration of uh, these ions in the phloem also determines the mobility, that how much mobile that element is. So you can see a huge difference in the concentration of potassium in the phloem and in the xylem. Then phosphorus, you can also observe the difference, chloride, sulfur, calcium. Now the calcium is perhaps the only inorganic ions which is more in the xylem sap as compared to the phloem. The reason might be that because there is a apoplastic movement of calcium into the xylem, and that make it a very high concentration in the xylem. And the mobility of calcium is very low. The mobility of calcium in the phloem, that is very low. So you might have observed while studying the deficiency symptoms of different ions. The ions which are not mobile in the phloem, which their concentration is very low in the phloem. So the deficiency symptom of that element occurs earlier on the root, on the young leaves, the growing tips. The calcium is one of them, because if there is a deficiency in the soil, so that will directly affect the growing parts. In general, the most of the ions which are mobile, so they will move from the lower part to the younger part for the survival, because these ions are required more in the growing part, so that's why they will move through phloem toward those parts which are still growing. But in case of calcium, you will see the direct deficiency symptom in the younger leaves, because their mobility is very low and their concentration in the phloem is very low. Magnesium, again, high in the phloem. Sorry, the sodium, it means the sodium is also not the only, although it's not a nutrient, but it is present in most of the soil and its mobility is also very important. So it means its concentration in phloem is very high. It means the loading into the phloem is very efficient. It is using the pathways, different pathways. We'll see that how it is loaded, how the ions are loaded into the phloem in the next slides. Then you can see the iron, zinc, manganese, all these are higher in the phloem. So this is the comparison of xylem. In the earlier slides, although we talk about the high concentration of organic compounds in the phloem, but you can see here that the inorganic ions, so they are also very high in the phloem. So the phloem loading, so loading of course is very important, and we know that it's a living cell. It's like a living cell. When we say living cell, it means it is covered by a living membranes. So when we say membranes, bilipid membranes, so it means the movement of ions and the bigger molecules that is not possible through the lipid bilayer, and they require transporters, as the transporters were required at the time of uptake, because the uptake in the root epidermal cells, that is also dependent on the transporters or proteins present in the lipid bilayer. So apoplasm by sucrose ion, sucrose H ion importers encoded by membrane of SUC gene families such as AT, SUC2 from Arabidopsis. So this is a gene family, and the transport is a co-transport or sim transport. So as we have earlier seen in the case of carrier, said that there is antipode, there is sympode. Because most of the sucrose, it is transported through sympode with H ion. So H ions, any uh, variation in the H ion concentration will affect the transport of sucrose concentration. So one of the evidence which we have observed that the potassium has a very significant role in the production of H ions, the protons, because the potassium activates the proton pumps and the protons are produced in the cell. And these protons, so they are utilized for the transport of sucrose in the from the leaf cell, because the sucrose is produced in the leaf cells, from leaf cell to the other parts. The other parts is the reproductive parts and the twig and the roots itself. And one of the deficiency symptom of potassium deficiency, one of the deficiency symptoms of potassium, so that is you may uh, observe the dull spots on the leaves of the plants, so which shows that there is a lack of transport of sucrose from leaf cells to other parts. So that is basically the excess of sucrose, because the sucrose is being produced there, but due to the deficiency of potassium or due to the production of protein, their loading into the phloem is very slow, and there is the accumulation of sucrose in the leaf cell. Specific transporters for sorbitol and mannitol have also been reported. A multitude of genes encoding transporters that can potentially load amino acids, urides, and short peptides. So it means the transporters of these organic compounds are also important for the phloem loading. So it means that they cannot be moved through the lipid bilayer efficiently. For nitrate NRT1 family, transporters are used. Members of ZIP family are thought to transport zinc into the phloem, and the iron, manganese, zinc, and copper are probably also loaded into the phloem by YSL proteins. So in general, these elements are transported to zinc tissue as a metal nicotinamide complex or in association with small proteins. So these are in association or in chelation form. They are moving in the phloem in many cases, but they can move as we know that these heavy metals are the micronutrients, so they prefer to work in the chelation form. But on the other hand, the macronutrients, so like the potassium, so they prefer to move as the ions independently. And one thing you might have observed here that the many transporters which are used for phloem loading, so they are the same family is also used for the transport of ions into the cell and then ultimately into the xylem sap. So that was how the loading of ions into the phloem occurs. So we know that there are specific transporters for specific ions. If we just talk about the nutrients, mean the inorganic ions. So they have specific transporters, the calcium and the potassium and the magnesium, and so the sum of the transporters they are also being shared by different ions. And the sodium, which is not an essential element, that also share the transporters of potassium to move or to be loaded into the phloem, similarly as it was using the potassium channels to get into the cells in the root epidermal cells. Mobility of the ions, if we just talk about the inorganic ions, the potassium, magnesium, phosphorus, sulfur, uh, and organic and chloride, sodium. So they are highly mobile in the phloem. When we say highly mobile in the phloem, it means they prefer to transport the ions where they are needed, either in the uh, growing tips or in the reproductive parts. So the deficiency symptom of these elements, which are highly mobile in the phloem, which never occur uh, earlier in the reproductive part, a young part, but they will occur earlier in the older leaves because the these ions will mobile from those leaves to the younger parts. Then iron, zinc, copper, boron, and molybdenum, they are intermediate, and the calcium that is very low mobile in the which has very low mobility in the phloem. So transfer of different ions can also occur in phloem and xylem directly. As we have mentioned in the earlier slides, perhaps it is a kind of a circulatory system that whatever is in excess in the phloem or in the xylem, it means, for example, we have a high concentration of any ions in the xylem, and this is not loaded uh, into the leaves or sink. So that will not accumulate in the xylem, but that will be transferred to the phloem, or at least the water molecule. So they will transfer from xylem to phloem. And you must remember that the phloem is living cell. So if any ion has to transfer into the phloem, so that must pass through the cell membrane. So they must use those kind of proteins, transporters for their mobility. So the transport, a transfer of ions is occur between the xylem and phloem, uh, and also the at the root level. So there is also exchange of at least the water molecule in phloem and xylem. Phloem versus xylem for transport of nutrients. So of course that depends on the, this is a very simple equation that what is the mass has been transferred. For example, if the total mass of 1 liter is transferred per hour in a phloem, so the mobility or the transport of nutrients will depend on the concentration. So what is the concentration of that specific ion in the phloem, that fluid, which is either in the xylem, either in the phloem, because that will follow this very simple equation. The total mass transfer will be equivalent to the velocity. किस speed से वो हो रहा है, and then the concentration of that specific ion. Uh, if the velocity is higher, the mass transfer will be higher. If the concentration is higher, of course, the mass transfer will be higher. And the rate of flow in the white loop one uh, is mentioned here. That rate of flow mean the velocity centimeter per hour. So that is much less in the phloem as compared to xylem. So in xylem, the flow rate, so that is much higher as compared to. So it means if even there is less concentration of ions in the xylem, so the total mass transfer may be the same, because although the concentration of the ions is low in the xylem, as we have mentioned in the earlier slides, but due to the high rate, high velocity, the total mass may be similar, or at least that is relatively more as compared to that in phloem. But of course, it depends on the concentration of those specific ions in the phloem or in the xylem. Uh, but one thing is clear, that the flow rate that is higher in the xylem, much higher in the xylem as compared to phloem. So this is the kind of, it's not nutrient mobility that the nutrient is mobile or not immobile. It's based on the days after leaf emergence, because the movement of ions, so that also depend on the age of the plant. Here you can see our days after leaf emergence, 1 to 12. So you can see that 2.7 and 1.4. nanomole per leaf, and there is a net change of 4. In the both, there is a plus. And then the days after leaf emergence 13 to 20, in the phloem, so there is a negative. And the net change is 1.36. So that gave us the transport. So as we uh, enhances the days after the emergence, so there is a change in the mobility of nutrients in the xylem and the phloem. So it means it's not constant. It depends on the age of the plant, because we will see in the last part of this lecture where we have the remobilization of the nutrient. So because with the age, the demobilization is also affected. The remobilization means the remobilization can also be mentioned as the mobility in the phloem, because the phloem is responsible for remobility of ions. And you can see that the most affected is the phloem, the mobility of ions in the phloem as we change the age of the plant. Xylem and phloem import of calcium, potassium, magnesium, calcium into the terminal bud and the young leaves. So and other aspects, that is the terminal bud and the young leaves. Terminal bud is the growing part, the very new growing part, the growing tips. So you can see here the concentration of these ions in the terminal bud in the young leaves. In the xylem, potassium is 3.9, in the phloem is 20.4. So it means the transport of potassium to the terminal bud is through phloem, because there is a much difference in the concentration. Sorry, in the transport. And then the magnesium, you can see there's high in the xylem as compared to phloem. And in calcium, of course, it's very low in the phloem. So that the clear what we have discussed earlier, that the phloem is not transporting calcium. So if there is a deficiency in the soil, so that will be appeared in the younger leaves, the tips. Uh, the scenario is different when you have the youngest leaves. So the youngest leaves mean these leaves has been developed, and you can see that the potassium is almost the same in xylem and phloem, because uh, there is not much requirement of that remobilization of potassium, because the leaves has been expanded. And then magnesium is same as phloem. It's not very much mobile as we see the potassium, but to a certain extent there is mobility. And again, the potassium, calcium, you can see here that there is a difference. So it shows that there is a variation of phloem and xylem transport of different ions in different parts of the plants. So retranslocation or the cycling of nutrients, so that is very important for the plant sustainability. If we don't have such kind of things, so then there are a lot of chances of toxicity in different parts. Uh, Here there is a example of white lupin and castor bean. The concentration of potassium, sodium, magnesium, and calcium. And the sodium is very important for such kind of retranslocation, because it exists in the system, and if there is not retranslocation, so there are chances of more toxicity. If we talk about the leaves, uh, the this shows the proportion of total uptake in percent. Sorry. So 96% to the leaf lamina of potassium, that is through xylem. 96% is through xylem, and export from here, that is 72%, that is through phloem. So there is then the net difference. Then sodium 45% and 33% is exported through phloem, magnesium 33% and 25%, calcium 29% and 12%. You can compare with the castor bean. So the scenario is almost the same, but you can see some difference like the calcium. So that difference is much higher in the castor bean as compared to white lupin. So there is a retranslocation and partitioning, but the species, the different plant species has different kind of mechanism for that. It means the retranslocation is not same. So that is also a one sign of the what you say the resistance of any. For example, if we have sodium here, and you can see the castor bean, there is a very low difference of import via xylem and export through phloem. So it means whatever is coming, it's going through the phloem. So whatever is coming from through xylem, mostly is thrown out through the phloem. So it means they are more resistant. Roots you can also observe here. To roots via phloem is 59%, and the cycling through roots, that is 39%. And the root to roots via phloem, the sodium is 33, then the magnesium is 20, calcium is 9, then in the castor bean is 85, sodium 9, magnesium 15, and calcium 1. So you can see here that to roots, the ions they are also flowing through phloem, because we know that the once the ions get entered into the xylem, so they have to move up, because the xylem is a non-living body, and then in the up part, so they may be retranslocated to the phloem, and then they are being utilized in the roots. तो जो roots की तरफ है movement है different ions की phloem है, mean the phloem is more responsible for the provision of ions in the root cell as well. So generally we can consider that because the roots they are in the soil, so they are just taking nutrients from there. Of course they are taking nutrients, and their nutrients are being utilized in the root cells as well. But that may not be sufficient, and even then the phloem transport, so that is important. So that was all about today. We will start the remobilization of the nutrients in the next lecture, inshallah.